Let's discuss drug antagonism. Antagonists, these are the agents which binds with the receptor and prevents the action of agonist on the receptor or the subsequent response, but they do not have any effect of their own. Thus, the intrinsic efficacy of antagonist is zero. For example, atropine, propranolol, naloxone. The combined effect of drug can either increase or decrease the overall response. This depends on the nature of their interaction. Drug antagonism refers to interaction between two or more drugs where effect of one drug is decreased or blocked by the presence of another drug. This interaction can occur at various levels such as pharmacokinetics, which includes absorption, distribution, metabolism and excretion of a drug, and pharmacodynamic that affects drug's mechanism of action. There are various types of antagonism: physical, chemical, physiological and receptor antagonism, which are further classified as competitive and non-competitive antagonism. Physical antagonism involves interaction between the drug based on the physical property. This involves process like adsorption. Here, one substance binds physically with another and prevent its effect. For example, activated charcoal. Due to its extensive surface area, it allows it to adsorb various toxins, including alkaloids. It does not chemically react with the alkaloid, but rather binds with them physically. This prevents its absorption. Therefore, they are used in treatment of alkaloid poisoning. Chemical antagonism occurs when two substances combine in solution, leading to inactivation of the active drug due to their chemical properties. For example, antacids. These are weak bases. Therefore, they neutralize the gastric acid and are useful in treatment of peptic ulcer. Chelating agents, they form complex with metals and are thus useful in treatment of heavy metal poisoning. For example, dimercaprol used in arsenic poisoning. Physiological antagonism. Here, two drugs act at different receptors or by different mechanisms on the same physiological system, but they produce opposite effect. For example, effect of adrenaline and histamine on the bronchial smooth muscle. Histamine, they act on H1 receptor present on the bronchial smooth muscle, which is GQ subtype, therefore increases IP3 DAG, which increases calcium concentration, therefore cause bronchoconstriction. Adrenaline, they bind to beta-2 receptor present on the bronchial smooth muscle, which is GS subtype, which increases cyclic AMP and cause bronchodilatation. This helps to relieve bronchospasm in cases of anaphylactic shock. In receptor antagonism, antagonist binds to the same receptor as that of the agonist and inhibits its effect. There are two types of antagonism: competitive antagonism and non-competitive antagonism. Competitive antagonism can be reversible or equilibrium type, or irreversible or non-equilibrium type. In competitive antagonism, both agonist and antagonist bind reversibly at the same site on the receptor. For example, atropine and acetylcholine on muscarinic receptors. Competitive antagonist binds to the same site on the receptor of the agonist, but they do not activate the receptor. They block the effect of agonist on the receptor. The binding of competitive antagonist is typically reversible. This means by increasing the concentration of agonist significantly, it can replace the antagonist from the receptor binding site. This is known as reversible antagonism or equilibrium type of competitive antagonism, which can be overcome by increasing the concentration of agonist. In the presence of competitive antagonist, the dose response curve of agonist is shifted to right. This indicates that higher concentration of agonist is required to achieve the same effect, but the maximum effect, that is efficacy of the agonist, they remains unchanged. In non-equilibrium type of antagonism, the antagonist binds to the same site on the receptor as that of the agonist, but binds it irreversibly due to formation of covalent bond with the receptor. For example, phenoxybenzamine is an irreversible antagonist of adrenaline at adrenergic receptors. Non-competitive antagonism. The antagonist binds to the different site on the receptor and prevents the agonist from interacting with the receptor. Since the antagonist is chemically unrelated to the agonist, it binds to the allosteric site or different site of the receptor but alters the receptor in such a way that agonist is unable to bind with the receptor. This prevents the effect of agonist on the receptor. In this type of antagonism, the antagonistic effect cannot be overcome by increasing the concentration of agonist. For example, diazepam and biquiculin are the non-competitive antagonists of GABA A receptor. In the presence of non-competitive antagonists, the maximum response, that is efficacy, of the agonist is reduced. This is because the receptor's ability to respond to the agonist is reduced. The dose-response curve shows decrease in maximum effect. This can be achieved regardless of concentration of the agonist. That is, there is a flattening of DRC seen in cases of non-competitive antagonism.